Equine viral arteritis is a respiratory and reproductive disease of horses. It's caused by a virus called equine arteritis virus, and it has a huge economic impact to the equine industry due to the occurrence of abortions in pregnant mares, death in neonatal foals, and most importantly, establishment of persistent infection in stallions. Persistent infected stallions, also known as carriers, can continuously shed the virus in their semen for extended periods of time, and the virus localizes exclusively in the reproductive tract. Currently, the mechanisms that lead to the establishment and maintenance of persistent infection in the reproductive tract are not well understood. My project is focused on elucidating the mechanisms that allow equine arteritis virus to establish persistent infection in the reproductive tract of the stallion. And for this, my hypothesis is that equine arteritis virus has the capacity to evade the host immune response and therefore establish persistent infection. To prove this hypothesis, I have four main objectives. The first one is to identify the target cells for this virus in the reproductive tract. The second objective is to characterize the inflammatory response in the reproductive tract. This will allow us to determine what are the immune mechanisms that are involved in persistent infection. In the third place, we would like to characterize the other components in the seminal plasma that might be associated with establishment of persistent infections, such as antibodies, and most importantly, seminal exosomes, which have been shown for human and other species to have immunosuppressive properties. And finally, we would like to perform some studies on primary cell cultures from the reproductive tract that will allow us to establish an in vitro model of persistent infection. Our preliminary results show that this virus localizes in a specific accessory sex gland of the stallion, which is the ampulla, but not in the testis. And this makes it a very interesting model of persistent infection because this is a unique feature of this virus. Our studies will clearly elucidate the mechanisms of establishment of persistent infection and also allow identification of novel therapeutics to induce viral clearance in carrier stallions. Thank you.